Hello guys, today we are going to discuss the thin layer chromatography. This is as similar to the paper chromatography, but it is the medium is going to be differ, where in paper chromatography we are going to use the chromatographic paper to run the pigment on it or the sample on it within the solvent. But here that base of idea of paper is going to change. Here it will be thin layer chromatography instead of the paper, where the thin layer will be used. Now here we will go through some points which are very important in thin layer chromatography. That is similar to paper chromatography technique, but it is going to differ with the following points compared to the paper chromatography. That is more convenient and less time consuming compared to paper chromatography technique used to separate non-volatile mixtures. The supporting material is a sheet of glass. This is the unique. The base can be the glass, plastic or aluminium foil. Now here on this sheet of glass, plastic and aluminium foil, the thin layer will be coated as the silica gel, aluminium oxide or cellulose. The adsorbent is known as the stationary phase. Always these silica gel or aluminium oxide or cellulose is called it as the stationary phase, and the solvent is the non-polar mobile phase. So that is what stationary phase is the polar one and mobile phase will be the non-polar one. The solvent which is going to run through the stationary phase after fixing the pigment or the sample. Now here the layer may be as thin as 250 micron meter for analytical separation and as thick as 2 to 5 millimeter for preparative separations. A binding agent such as calcium sulfate or gypsum may be used incorporated into the chromatographic media to facilitate firm adhesion of adsorbent to the plate. Now here whatever the silica has been used or aluminium oxide or the cellulose to prepare the thin layer on these base materials, in that we are going to add a proportionate calcium sulfate or the gypsum, which is going to hold a good bond on the base of this particular thin layers. Now here most commonly used of this particular thin layer is the Silica Gel G Merck, which is having again 13% calcium sulfate. So here what is that, it should be prepared immediately and it should be used immediately to prepare the thin layer. Another commercial adsorbent is the Silica Gel H Merck, does not contain calcium sulfate. Here we have to add a proportionate of calcium sulfate to prepare the thin layer. Now here you have understood two different silica gels, one is here, one is here. That is the Silica Gel G, Silica Gel H. Silica Gel G is used because it is already having 13% of calcium sulfate. Here it is used very less because it is not having any calcium sulfate kind of the binding agents. Now here, as per what discussion has been done in the previous slide, we are discussing the differences. In paper chromatography, these are the four features, and in thin layer chromatography, these are the four features which are going to be contrary with each other. One is, in paper chromatography, the separation is based on partitioning only. So it is where we are going to partition the component which is present in the sample, whereas in the thin layer chromatography, the separation can be achieved either by partitioning, adsorption, ion exchange, or molecular sieving phenomena. So here these are the four different separations which we are going to do in the thin layer chromatography. It may take up to 8 to 24 hours R&D labs where the timing is going to be considered, whereas in the thin layer chromatography is extremely rapid as the separation can be achieved within hours. This is also within the R&D labs. Now when you are going to compare this with the lab experiments done in the colleges, that is going to be very different. So here these are the analytical and preparative kind of the run over of the chromatography with the sample. An ideal concentration of sample is always required. So here perfect amount of the concentration is always required. Without that the paper chromatography cannot be done. In thin layer chromatography, lower concentrations are also sufficient in the mixture form which can be successfully separated. Whereas here, if it is a mixture form of the component where we have to separate more than two of the samples or the components, here there should be a required amount of the both the samples or the required amount of the sample to separate each other from on the proper chromatography. But here in the thin layer chromatography, low concentration of the sample is also sufficient to separate the mixture and to detect. Here you cannot use sulfuric acid on the paper. Let it be at any concentration, and you cannot use the high temperatures because the volatile or non-volatile components only, but because of their being very less in concentration, as the component has been run through the paper, it can disappear due to the evaporation. Now here, if you're going to consider in thin layer chromatography, corrosive agents like H2SO4 and high temperatures can be efficiently used to locate the separated compounds. So as the base medium which we are using, like glass or plastic, the other components, those are going to withstand, or the whatever the thin layer which we are going to use a silica gel, and with the component of the gypsum or the calcium sulfate, it is going to give the firm basement or the firm matrix to hold good the samples. So the matrix compared to here, it is going to have very high difference. Because of that, this is going to be the a large difference which is going to matter a lot. Here, mobile and stationary phase. Stationary phase is the one which we are going to use as the either as the paper in the form of the paper chromatography, or the thin layer for form of the sheet which is in the thin layer chromatography on the plastic or the glass. Now here, that is the stationary phase. Mobile phase is one where we are going to use the solvent which we are going to run through overall the paper chromatography or the thin layer. Now here, volatile solvents are liquids that vaporize at room temperature. So here we are going to use the solvents which are little volatile, which are going to evaporate at the room temperature. So here, for example, if you are going to consider the acetone is the volatile component which is going to evaporate or vaporize at the room temperature. So here, non-volatile solvents refer to the liquids that does not readily evaporate into a gas under room temperatures. Now here, the liquid or gas that flows through the chromatography system, moving the materials to be separated at different rates over the stationary phase. So here, having the volatile component as the solvent is good. When you are going to dip the thin layer slide or the medium into the solvent is going to run equally by separating the sample. Because of that, the sample is going to separate at the regular grades, because depending upon its attraction on the layer. Now here, when you are going to consider the mobile phase is always taken to be as the non-polar in nature, whereas the stationary phase is considered to be as the polar in nature. So here you can see the stationary phase remains fixed in the place and polar in nature, whereas the mobile phase can range from non-polar to very polar solvents. It is here very polar solvents is required in some. Now here you can see what is polar molecules occur when there is an electronegativity differences between the bonded atoms, that is the water, ammonia, sulfur dioxide, hydrogen sulfide. Non-polar molecules occur when electrons are shared equals between atoms of diatomic molecules, or when polar bonds in a molecule cancel each other out, that is carbon dioxide, benzene, calcium tetrachloride, methane, ethylene. Now here, when you are going to consider electronegativity differences, it is the partial charges. They are going to show partial charges where they can pull the component to any of the sides. When you are going to consider here ammonia, here the component, when it is going to run over this polar medium, whatever the sample, it can pull the sample at the any of the end, and because of that the separation of the molecule is not going to take place properly. So when you're going to consider the non-polar is going to run equally over the chromatographic paper or the thin layer chromatography. So here that is what the meaning. Now here, how to prepare the thin layer chromatography plate. Thin layer plates are usually commercially available with standard particle size. They are prepared by mixing the adsorbent. Now here, if you want to buy a commercially available, you can buy directly, but you also prepare it in your labs. Adsorbent mix as silica gel. Whatever the adsorbent you are going to buy, as we have discussed as the Silica Gel G Merck, like that, if you are going to take the small amount of the inert binder like calcium sulfate and water. Now here he has given the things separately, but if you are going to get or buy the Silica Gel G, there is no need of calcium sulfate, and but you have to use the water to mix it and spread it equally on the either the glass or the plastic side. This mixture is spread as a thick slurry on a unreactive carrier sheet. Unreactive carrier sheet is nothing but the usually glass is used. Thick aluminium foil can also be used, or plastic also can be used. The resultant plate is dried and activated by heating in an oven for 30 minutes at 110 degrees centigrade. Activated in the sense they perfect layer formation and the matrix formation has to takes place during this time of the 30 minutes. The thickness of the adsorbent layer is typically around 0.1 to 0.25 millimeter, that is 250 micrometer for analytical purposes, and around 0.5 and 2.0 millimeter of preparative purposes. Now here, this is the particular procedure where we are going to how we are going to run the sample. So here we are going to take a particular container, or the container can be like this, where it should have a sufficient space to place the thin layer chromatography sheet, so that it can get into that particular container, and further we should be able to close the container. Now here you can see this one is one. This is the schematic representation or depicting the sketched picture, whereas here you can see where the labs have used the containers like this. Apart from this, there are different shapes of the containers depending upon the availability in the lab, but here you can take whatever is available in your labs. You can use the measuring cylinders also, which are quite good to place your thin layer chromatography sheet, or you can use any conical flask or the beakers which are small in size, but you should be able to close with any of the lids, so that you can perform this particular steps. First is that a small amount of an appropriate solvent has to be taken. So here, whatever the experiment you are doing of the thin layer chromatography, for that you will be having a appropriate solvent for that to run over that particular thin layer chromatography. That solvent we have to take it earlier itself before placing the thin layer chromatography sheet. So what we are going to do, we are going to take this solvent is poured into the glass beaker or the container in which we are going to run the thin layer chromatography to a depth of the less than 1 centimeter. So here whatever less than 1 centimeter means to the bottom, from the bottom of the into the inner side of the container to that above 1 centimeter should be covered with the solvent. A strip of filter paper is put into the chamber so that its bottom touches the solvent and the paper lies on the chamber wall and reaches almost to the top of the container. Now here, before placing the thin layer chromatographic sheet, we have to place the chromatographic paper within that particular container, and we have to, after pouring the solvent, and we have to close the lid so that the container should be closed with the cover glass or whatever the lid is present for few minutes to let the solvent vapors ascend the filter paper and saturate the air in the chamber. So if you failure to saturate the chamber will result in poor separation and non-reproducible results. Now here the separation will be poor and the results will be errored. Now before placing the thin layer chromatography, we have to do whatever the earlier steps which we have discussed. During that time, when we are going to leave the container to get saturated with the volatile solvent or the eluent, during that time we have to take the thin layer chromatographic sheet which we have prepared already, or the plate which we have prepared, and we are going to take the capillary tubes, and we are going to take the small spot of solution containing the sample and apply it to a thin layer chromatography plate. And remember, in many instances we are going to use the solvent as the medium to dilute the sample. That is the thing which we are going to do. So before placing these spots, as we are seeing here, before placing these spots we are going to do that little dilutions with the solvent, and we are going to place the spot repeatedly at the same point with the help of the capillary tube. So here we are going to keep this spot exactly about to the 1 centimeter, about to the 1 centimeter, in the sense to the 1 centimeter there will be the solvent. So about to the 1 centimeter of the thin layer chromatographic plate we are going to keep the sample dots whichever is required. Now here, when you are going to, after placing the sample dots, you are going to leave that thin layer chromatographic plate to dry for some time. After drying, the solvent is allowed to completely evaporate off to prevent it from interfering with sample interactions. If a non-volatile solvent was used to apply the sample, the plate needs to be dried in a vacuum chamber. I hope this point is understood. This step is often repeated. It has to be often repeated because the enough amount of the sample has to get concentrated on this particular point. Then only there will be the enough amount of the sample within this point to get separated all over the thin layer chromatographic plate after it is placed for the further procedure. Different samples can be placed in a row of spots. Now here you can see, you can keep the different samples. Certainly, if you are going to consider, there will be a reference to the sample which is used. For example, one sample is used to run over the chromatography. For that there will be a reference. If there are the two samples used, there will be the two references. A sample can be a mixture of two different runs. So for the two different runs also there can be the two different references. So because of that there can be the reading of the 3 or 4 dots depending upon the requirement of the dots can be sent. Now here you can see that here this is the two different dots, and here also there is one which has been kept, but the sample is appearing here. The samples are appearing here, the separation which we can be detected. Now here if you are going to consider here it is only a single sample which has been used, there is no reference. Here also it has been used, there is no reference, but it is for chlorophyll and whatever the leaf sample has been taken. Here this is also the same. This is from the spinach, and this is the fresh spinach, and this is for the frozen spinach. So you can see the separation. Now further, the plate is then placed in the chamber. Once the sample is got dried with the enough amount of the concentration, it has to be placed here. Here you can see the solvent is below to the sample line. Now here there is the difference of 1.5 centimeter to the bottom of the container to the line drawn on the thin layer chromatography plate. So now the sample has started to run over the thin layer chromatography. With the run of the solvent, the sample has got separated. Now you can see the amount, depending upon the amount of the attraction. If the attraction is more, the solvent is going to run only to the less distance, whereas if the attraction is less, then the solvent is going to run to the high distance. Now here, as depending upon, the solvent has to run only till to the edge of the thin layer chromatography, but before to the front, it has to be stopped at the 1 centimeter below to the edge of the thin layer chromatographic plate. Now then you have to take and you have to evaporate, and after that you have to mark the separated samples, and you are going to further visualize it. Now here, if you are going to consider visualization, in this the visualization will be with the naked eyes, as these are the pigments which can be seen, whereas in some components the visualization is not going to take place. During that time you have to use some plates which are pre-coated with phosphor, where the phosphor is going to glitter or refract as the radium under the UV light. So here, when you are going to run on those plates the samples which are going to run on the thin layer chromatographic plate with the phosphor is going to show the opaque nature depending upon the thickness of the swabs or the dots. Now here it is going to show the opaque nature. Now here you can see the opaque nature depending upon the concentration of the whatever the separated sample. Here also you can see. So that is what. So with the help of the UV chamber at certain nanometers, these particular dots can be marked with the help of the pencil, so that after you take out the thin layer chromatography plate, you can read the run of this particular separated samples. So alternative plates can be sprayed or immersed in the chemicals after elution. Various visualizing agents react with these spots to produce visible results. Now here, apart from this UV kind of the technique, there are two different techniques where we are going to treat them with the chemicals. So here various visualizing agents can also be used to spot the whatever the separated samples has been present on the thin layer chromatography plate. Now here, this is what the thing. If the used sample is having the pigment, then the pigment is going to separate this particular in this particular form. So here the sample mixture of different compounds travel at different rates due to the differences in their attraction of the stationary. So the differences in the attraction of the stationary phase, and because of the differences in solubility in the solvent. Now here, whatever the sample we use, that is going to have different solubility of that particular nature of that particular component or the molecule present. Because of that, the sample is going to get separated. That is what even I have spoken. That is, if there is a high attraction, there will be the low separation, low distance traveled. If it is the less attraction, there will be the high distance traveled. By changing the solvent or perhaps using a mixture, the separation of the components can be adjusted. In the sense, if you are going to use one solvent, if it is going to give one RF value, if you're going to use the different solvent, that is going to give the slightest difference in the RF value. So here that is what the reference with. Chemists often use TLC to develop a protocol for separation by chromatography and the thin layer chromatography to determine which fraction contain the desired compounds. So here thin layer chromatography is very significant. So the chemists are using to separate the products are present within the chemicals, the components present within the chemicals. So they are going to use it to determine which fraction contains the desired compounds. So here this line is very much informative, that is which fraction contains the desired compounds. So during the separation of a particular sample, they are going to use the reference, and with the help of the reference they are going to study the fraction containing the desired compound. Now RF is the migration distance of substance, migration distance of solvent front. Now here when you are going to consider, if this is the spot of the sample and if this is the spot of the end of the solvent running, so from the spot of the sample to the from the spot of the end of the solvent running is the distance of the solvent front, and from migration distance of substance is the from the spot of the sample placed and till to the first separation which has been formed till here, that will be first, and second will be the from the spot to the second component ends, third will be the from the spot of the sample placed to the third. So that those are going to be placed in the numerator. So this will be divided by this, and whatever the value we are going to get, that is the retardation factor, that is the fraction of an analyte in the mobile phase of a chromatographic system. So this formula remains same both for the paper chromatography and also the thin layer chromatography. So here this is what the thin layer chromatography, where I have made this as a very brief one. I did not dig more points, and I hope I have given much information. So I don't want to make a slide or the video log of a hour or more. So I have tried to very simple manner, and I thank you guys. If you have any doubts in this video log about thin layer chromatography, you can come across and put it in the comments, and also if the any wrong has been spoken, you can ask me, and I am ready to reply. And if you have any new experiment has to be spoken out on the video log, please type out the title and be very clear with the experiment, because within a title there will be the lot number of the procedures and the components will be used. As specific to your requirement, please give some details so that I can come out and prepare some video logs so that it will be helpful for you, because many are going to give the titles, but under the titles there are many kind of the what you called subtitles will be present to perform the experiment. So please be very specific so that I can come with your requirement where I can accomplish or serve with your request. Thank you very much, and be tuned to Big Book, the Learner's Big Book, so we can help you more compared to anybody else. Thank you very much, and help us to spread our word.